Good evening, everybody. We're going to start our program. I'm Omai Garner. On behalf of AACC, I want to welcome all of you to this evening's program called Winning Organization-Wide Support for Laboratory Proposed Acquisitions. I'm the Assistant Clinical Professor at the UCLA Health System, as well as Associate Director of Clinical Microbiology and the Director of Point-of-Care Testing for our health system. I will be your moderator tonight, and I'll start the program with a brief overview of some of the challenges and strategies I think that we face as lab directors for laboratory purchasing. But before I begin, I have a few announcements. So first, I would like to thank Roche Diagnostics for supporting this event. Roche supports many AACC programs, and we greatly appreciate this support. There will be an opportunity to ask questions to Dr. Colbert and I as a panel afterwards. So if you guys can hold your questions to that point. And then finally, there are continuing education credits available for this, and you will receive an email tomorrow from AACC for everybody that's registered with instructions on how to claim credits. So I'm going to begin with a talk. The title of my talk is Purchasing New Lab Equipment, Challenges and Strategies. I have a few disclosures for honoraria. Oh, oops, I got to hit both buttons. Sorry about that. There we go. So I have a few disclosures for honoraria, research support, and consulting for companies that would be relevant to this particular talk. So just as a little bit of an outline, so I'm going to get kind of a brief overview. I want to talk a little bit about laboratory evaluations, not the purpose of this talk, but they go into how we decide on what we're going to bring in as a product, analytical and both workflow. And then I kind of want to dive into some of the purchasing options that are available when you have a piece of laboratory equipment that you want, right? So is it capital equipment? Is it lease? Is it reagent rental? What are some of the advantages and disadvantages to all of those systems? And then what I really want to talk about are our hospital committees, right? The deciding committees as to whether or not we get the equipment that we want. There's a lot of acronyms there, VAC, ROI, SRC, and we're going to go into a lot of those. And just the caveat here is that this is what's relevant for the UCLA health system, but it may not be what's relevant for all systems, right? So I'm kind of giving you a snapshot and what you may see a difference between what I talk about and what Dr. Colbert talks about in the way that our systems work. But in general, we kind of see this is how this works. So I want to start out with a rep showing up at your facility, and they have the new best assay in the world, right? I was pretty agnostic and tried to pick the most general picture of lab equipment I could possibly find. But this rep comes in, and of course, they've got 100% sensitivity, 100% specificity. The turnaround time is instantaneous, and the machine never breaks down. So, because of this, I'm going to put this as our mystery magic box kind of in the bottom corner. So what do we do then as lab directors when we hear this? So the first thing, of course, that we do is a clinical lab evaluation. So I wanted to give you kind of an example of what we look at as a clinical lab evaluation at UCLA. And so here I'm taking two products that'll go unnamed. We'll call them New Assay 1 and New Assay 2. And these are two assays that do flu testing. We were evaluating these flu testing assays within our laboratory to think about a change in flu testing. So in order to make that work, what we ended up doing is taking 150 de-identified nasopharyngeal specimens that I had stored at minus 80 degrees within my microbiology laboratory, and I wanted to test them on the new platform and compare those to the results that we got on our PCR, which we'll call our gold standard, right? And so I took 84 positive flu A's, 16 positive flu B's. I wanted to do a little bit of analytical specificity, so my negatives weren't just truly negative. They also contain other relevant viruses. So I ran about 75 rhinovirus positive and 25 of viral transport media alone. And this was just a straight one-to-one comparison of the two platforms, right? Now I divided them up through sensitivity of amount of nucleic acid that's there. So for those of you not in molecular biology or in microbiology, that CT value is a range of the amount of flu virus that was in the specimen. The lower the CT value, the higher the amount of flu specimen. So we tried to organize it into kind of specimens that were spread throughout. And then we compared the two devices, sensitivity, specificity, invalid rates, which are important. And as you see across the board, comparing New Assay 1 and New Assay 2, they had about the same percentage of invalid rates, which was nice. The specificity was 100%, so they were very good at calling, but the sensitivity we saw changes. So for new assay 1, the sensitivity was very close to 100%. The problem for new assay 2 was that it was closer to 70% as compared to our gold standard. So from a clinical microbiology laboratory director, I've got a good device. But oftentimes with devices, especially because this is in the point of care world, you don't just need to think about the sensitivity and specificity of the device. A lot of it's going to be about workflow. Right? And so a workflow analysis is critically important to knowing whether that device is going to work in your lab, in the outpatient setting, wherever it's going to be. And so the way that we typically do a workflow analysis is we come up with some level of survey, and then we bring the tester in-house to do a level of an analysis and then have an opinion on whether or not they like that particular instrumentation. So here, the blue bars are new assay 1, the orange bars are new assay 2, and you can see we have various parameters, right? Is the instrument easy to load and operate? Are the instructions for use easy to follow and memorize? Is the graphical user interface easy to use? Can the test be performed in a certain number of steps? So various criteria, and then at the end of that, we're going to choose which instrument is going to be better from a workflow perspective. Now, from a lab director perspective, it has to meet sensitivity, specificity criteria before we even get to this point, right? But if it meets it, then we're going to do this workflow analysis. So let's say everything is going really well. Right? This new best assay in the world method not only meets your sensitivity and specificity requirements, but it also is great to your laboratory. Your laboratorians love it. You want to move forward with it specifically. So the next thing you want to think about is how you're going to buy that particular assay. And there are different ways to buy an assay within our hospital system. But when we're thinking about an assay, we're thinking about the whole package, right? We're thinking about the instruments, we're thinking about the reagents. So let's first just focus on the instruments, because the instrument for a lot of the best new assays in the world are very, very expensive, right? So how are we going to justify this, or how are we going to purchase it? So the first thing you can do is try and get your very expensive instrument on the capital equipment list. So this is a list of items included in the upcoming fiscal year budget for the purchase by the hospital. What's really nice on this is that the hospital has a pretty big box of money. My hospital has a pretty big box of money. And so you can put fairly high price items on that particular list, right? So it can cost $250,000 and potentially it can be paid for on that. The challenge there, though, is that that capital equipment list is typically reserved for items that are critical to lab performance, right? So when you think about something that your lab needs to survive, centrifuges, incubators, freezers, refrigerators, all of these things would take priority over your new best assay in the world. And so what we can see is that you can get bumped off the list if a bunch of freezers break down, right? Because you got to have those -80s. So suddenly you were on the capital equipment list and now you're off the capital equipment list. The other issue for us at UCLA is we share the capital equipment list amongst the clinical lab. So not only am I competing with freezers going down within the microbiology laboratory, I'm competing with freezers going down in the chemistry laboratory. So maybe everything's working out wonderfully in microbiology, they lose 9 freezers in chemistry, my best new assay in the world just got bumped off the list again. So we have to consider that in thinking about how we're going to think about paying for it. So what are some of our other options in paying? So we could lease, right? Lease is pretty straightforward, especially with expensive instrumentation. You take the price of that instrumentation and you pay by rent basically over time. And then sometimes at the end of that lease, be 3 years or 5 years or whatever it is, you can buy that instrument at a reduced cost. So let's say maybe at the end of that lease, you have a $1 buyout. Right? This is a good way to purchase a piece of equipment, but there are some issues in this as well, right? The first issue is that you've locked yourself into a fairly long-term contract, right? If it's 5 years, if it's 7 years, as we all know, things move real fast in clinical lab medicine. So what if in 2 years, just like maybe your iPhone or something like that, that new best assay in the world is obsolete? Right? And now a different company having the best gold standard new best assay in the world is knocking down your door, but you can't switch it over because you're tied into that lease. So that represents a challenge as well. The third way that you can buy something is through reagent rental. You can kind of think of this as a modified lease, right? So you take the cost of the instrumentation, you spread it out over the cost of the reagents with an added fee on top of each reagent to pay for or lease the instrument. So this works out well if the cost of the instrument and the cost of the reagents are high, right? Because then you can add that money on top of the reagents. The challenge here for this is that if you have a piece of equipment like a MALDI-TOF is a mass spec that we use for bacterial identification. Very high instrument costs, really low reagent costs. Right? So if I'm only spending 20 or 30 cents on a reagent on a $250,000 machine, it takes a whole lot of additional 20 or 30 cents to add up to do a reagent rental on that. So for a lot of things, reagent rental can't be used. The other issue, I think, with reagent rental is that you need to be able to accurately predict the number of reagents used over the period of the contract. Sometimes you're very good at this, sometimes you're not very good at this, right? So at UCLA, we see this average increase in patients of about 3 to 8%, and we kind of monetize that over all of our testing, but sometimes we're wrong. So if we're guessing and we're saying, I'm going to use 100 reagents and our reagent rental is on that, but I only use 50 reagents, I end up owing the money to the company after that. If I guess the other way, right? So I guess that we're doing 100 and we did 500, well, now I've actually overpaid for that piece of instrumentation. So really, to use your reagent rental, you're going to want to know exactly how many reagents you're using, and that can be over the life of the contract. So sometimes it's hard to predict over 3 years or 5 years. But I would say regardless of all of that, whether you choose capital equipment or lease or reagent rental, and a lot of times a company will provide you numerous options, you have something that you've clinically evaluated and you like it from a workflow perspective, you want to move into buying that new best assay in the world. So you have to account for costs of instrument and preventative maintenance, reagents, and people to perform the tests, and you can compare that directly to the cost of what you're currently running. Right? So you're kind of looking at apples to apples. And what we find at UCLA is if that's budget neutral to your individual lab budget, so the amount of money that I spent on whatever the new BAS assay is equivalent to the amount of money I was spending, the answer is you can just buy that, right? Because to the hospital's budget, it doesn't look like anything happened. Right? You just transferred the money over. So that's very exciting, and that's why we often try and push the companies that have the new best assays in the world to be able to meet whatever my old price was. The challenge is we're dealing with new technology, right? And new technology is often more expensive. And so there is going to be some added cost. Now, at some institutions, the added cost can be under a threshold that you can still buy it. And we're going to talk about what that threshold is at UCLA. But if it's going to cost any more to the lab budget, then we have to face, oh, you know what, I forgot. Sorry about that. I'll get it right. So we have to face one of these committees, right? And so the challenge of one of these committees is that we have VAC, the Value Analysis Committee, ROI, the Return on Investment Committee, and that's mainly what we're going to focus on. So what is a Hospital Value Analysis Committee? So who this committee is a committee consisting of physician leadership, nurse leadership, and hospital administration. I think it's good to note here, as a room full of laboratorians and people interested in the lab, most of the time there's not laboratory leadership on this particular committee, right? So you're already having to define lab to people who aren't used to talking about that. What role do they play? So the role of the Value Analysis Committee is to define value and decide exactly what's going to get paid for out of that box of money in the hospital budget. The challenge is that that box of money is limited and you're competing with every single person across the hospital system. All the initiatives have to go through VAC. So now my best assay in the world is competing against the new best surgical tool in the world, right? And everybody has something new. So we're going to need to express our value as compared to all those other things. So why do these things exist? I think the reason why things like VAC exist is to protect patient safety and also improve patient care. A lot of things have been brought into hospital systems, and because there wasn't a committee, maybe they weren't the best things, maybe they were even harmful to patients. So it protects from that. It also leads to some transparency, so at least anybody who's trying to get something purchased can know what the process is going to be, and that process is going to be standardized, right? There's some impartiality, but I would argue it's not that impartial if there isn't any lab leadership on there. So the impartial thing really kind of depends. And I highlight in bold is cost control. Cost control is probably the most important. It's probably only second most important to quality patient care, right? So as healthcare gets more and more expensive to pay for, hospital systems have to survive. And the way that you survive is by counting your dollars. So what they really want to know from a VAC perspective is, how will your new initiatives save the hospital money? Or when are you going to return that money that's spent to your hospital? So what I wanted to walk through, I've actually pulled directly from UCLA's VAC list. So you get a list of the things you need to fill out to be able to go to VAC. So let's walk through some of these and think about what it is they're actually asking for. So to get on the VAC list at UCLA, you're going to need to identify your product and manufacturer, which is very straightforward. But the first thing you're going to need to list is all companies developing a similar product. Now this is interesting, right? Because you got your new best assay in the world, but you may have your second manufacturer new best assay in the world. What they want to know is who else that is and what does that cost? Because eventually they may decide, yeah, you can get the new best assay, but we want to go with this manufacturer because they're 50% less. So they need you to provide that information. Also, financial conflicts of interest are important from anyone involved. The next section is a clinical intended use and impact of the product section. And this is where you get to kind of describe the impact total to the hospital system in as large a way and in as non-lab centric, outside of lab understandable way as you possibly can. And there's a science to this. There's a nuance to this. Because if you lead in this area with something that they don't understand, it's over at that point. Is there a product in-house performing a similar function, and why do you need to go to something new? What other departments will use and be affected by the product, and this is a big deal, and have you told them about it? So if I'm going to VAC and I'm saying, wow, this is going to have a massive impact on the emergency department, this new lab test, and the emergency department's like, I've never even heard of this, right? That's an issue. That's a point against you for VAC, right? Of course, they want projected annual usage volume and approximate costs. And what's interesting here is they actually define a different line here from a money perspective. So they ask you straight out, will the projected annual dollars spent exceed 100,000? And so what this means to UCLA is there's a couple of different ways they look at it. They look at projects under $100,000, and then they look at projects over $100,000. And so where you are on that line is going to matter in how you do with VAC. Is the project patient chargeable? And this is interesting because remember, you're comparing against like surgical tools and things like that. So are there reimbursements? Do those reimbursements even matter? Is it inpatient? Is it outpatient? All of that data. And then is it urgently required? Right? Because if you're comparing against something that's going to go in the Ebola lab, let's say, and if we're having an Ebola outbreak, well, that thing's probably going to get funded before your idea because it's urgent, it's emergent. And you want to be able to explain that. They want to know whether or not you've evaluated the product. So remember when we looked at it early on, they're going to want to take that data in. They also want to know whether or not there's published literature. And the published literature isn't just sensitivity and specificity, right? That's what we think about from the lab. They're really interested here in outcome studies, right? When you say your new best assay in the world is going to have this clinical impact, is there anything at any other hospital system that has already proven that? And you can use that data with VAC. Additionally, they ask you to provide all supporting documentation, and what this means is personal letters from vested physician groups. So if you say it's going to have a positive impact on the ED and affect admission rates, then the head of the ED needs to write a letter that says exactly like that, right? Same thing with infection prevention. If infection prevention says this is going to have a huge impact on antimicrobial stewardship, then that physician needs to write a letter for that. Then at this point, they have this interesting quote, and I just wanted to read this to you. So you filled out this entire form, and you're getting ready to present it to VAC, and they say, the Value Analysis Committee requires the clinical budget responsible director/manager be informed of your initiative, and your department or division chair chief review and approve your request prior to committee consideration. This means take this paperwork to your division chief in your department before we see it. Because if they don't sign off on it, there's no reason for us to read it. Right? So they want to put another check and balance before they use committee time. And here they say all initiatives that are approved in greater than $10,000, so approved by your department, will require Value Analysis Committee review for final decision. So they laid out the lower limit threshold at UCLA, which is currently $10,000. Anything below $10,000, you don't need to go to VAC for. Your division can approve that and you can spend it. Anything above $10,000 is going to have to go to VAC. So the committee meets once a month to be able to renew the initiatives, and then they'll bring in the person who presented the initiative to be able to defend and answer questions. This is also a challenge. If you send the wrong person to this meeting, you can have a great proposal, and you may not get funded because you're speaking to non-lab people, right? And so in speaking to non-lab people, it's about having as simple and straightforward language as possible, and deciding how at your own institution being able to tell that particular story is very important. So I wanted to quickly run through some examples. We're going to get a very detailed example of this process, but for us in the laboratory, one of them was Malditol for bacterial identification. We talked about this a little bit. High capital costs, low reagent costs, and we were actually able to justify this just based on lab savings alone. So what this meant to recoup the money to the hospital that they were going to spend out in capital equipment is we had a 3-year business plan, and we were going to save money on the automatic biochemical identification cards. So this basically paid for itself. So it was great. Now what was interesting is after a year, they came to us and said, show us a third of our money back. So you have to be ready for the committee to show back up, because they can pull what it is that you're doing if you aren't making the progress that you said you were going to make. So the stakeholder there is the lab, right? But then if we look at something else like that's going to be a cost loser to the lab, like PCR for TB on sputum specimens. The challenge there is that PCR is way more expensive than something like an acid-fast smear. So it's difficult to justify on lab costs. But you can make a business plan based on isolation room costs, right? So potentially your PCR can reduce isolation because negative PCR results can be used to release patients, and then you save that money per day. So your people in that are lab people, but also infection prevention are your champions there. So we're able to justify that cost in that way. And then finally, even though it's controversial right now, we'll land on C. diff testing. So Clostridium difficile testing we're doing by PCR. This was an initiative we brought out 5 years ago. At that point, it's also more expensive than the thing it was replacing, but we can look at drug usage, right? So reduction in oral vancomycin usage for patients that were C. diff tested negative could potentially lead to the cost savings, and there our stakeholders were lab and pharmacy, right? So these are just ways to go about getting at VAC and proving the value, or at the very least breaking even. So interestingly, for years, this is how we went at UCLA. And then recently we had a project denied to us where we had a good business plan that was able to show that we were going to recoup our money, and we were informed that there was a new committee. And so the new hospital committee was called the Return on Investment Committee. And so this was a hospital committee that was actually just made up of hospital administration, so the C-suite, and finance. And that was it. And so what they said at the C-suite and finance was that it's not good enough just to make your money back. We need to make $1 million. So your new initiative must yield an overall gain of $1 million over 5 years. Now I take this as good and I take this as bad. Right? It's bad because it makes it a higher challenge, but it's good because it gets us laboratorians out of the lab, right? So we need to go to other areas of the hospital. Look, we're not going to save $1 million over 5 years in the laboratory, right? But if we can think about patient rooms, if we can think about medication, the effect of every single new test we bring in has a scatter effect throughout that hospital. So now it's driving us to be able to prove that, gather those people together, and then present that. So an example of this from 4 years ago, so before there was an ROI committee, but we did a mailer program for the Department of Pathology for fecal occult blood screening, right, for colon cancer. Send out the mailer, it ended up costing $50,000, but then when we're doing our study with the GI team over time, we've seen an overall reduction in stage 4 colon cancer. That's very expensive, right? So we've now been able to show that $1 million fairly rapidly. So it's doable. It just kind of makes you think a little bit out of the box, and it's important, I think, for laboratorians to get out of their lab and show their value to the hospital. So I'm definitely taking that as a half glass full kind of way of looking at the return of investment committee. So ultimately, none of that may work, right? And you still may be told no, and a lot of that has to do with the inavailability of outcome studies. So you may think that your test is going to do this, but there's no proof that it's going to have an effect on things like ER admissions. So at that point, the alternative strategies, or what if the new best assay in the world still isn't approved, is to be able to go back to the manufacturer and say, let's design a pilot study. Right? Let's try and prove to our hospital system that this is going to save the money that we say it's going to save. And so let's bring the product in-house, maybe 6 months, maybe a year, and attempt to approve the cost savings to the hospital. So that's my real quick introduction, kind of on how we see costs, how we see cost savings, and what hospital committees exist. I want to thank Paul Colonna and Cindy Toy. These are the managers and the hospital side people who over the last 5 to 7 years I've learned all this from. And then, of course, Dr. Binder is our vice chair of the department. So now I get to introduce our next speaker. So Dr. Carissa Colbreth, who is going to discuss her experiences with planning for and implementing major laboratory purchases in her institution. Dr. Colbreth is the Scientific Director of Infectious Diseases at TriCor Reference Laboratories, and she is an Assistant Professor of Pathology at the University of New Mexico Health Sciences Center. At TriCor, her primary focus areas are clinical microbiology, parasitology, mycology, and infection. Her research interests include use of emerging technologies for diagnostics in microbiology, emerging nucleic acid amplification methods for rapid ID of bacteria and fungus, and microbiology capacity building in international and domestic resource-constrained areas. So please help me welcome Dr. Colbreth.
Thank you very much. So this is definitely an interesting topic. I don't know how exciting it is, but it certainly is interesting, and I think it's something that we all face in the laboratory, trying to figure out how are we going to get our capital purchases and these wonderful new technologies that we've all seen on the exhibit hall floor. I think everybody's walking away with a pamphlet that they're planning to take back to their administrator and say, I want one of these. And my goal today is hopefully we can help you figure out how to get that, okay? So we'll try to do this together. So just a little bit about our laboratory, because I'm going to give you some of the perspectives that we face. Every laboratory is going to be different, but this is the way that we've approached looking at major capital investment and return on investment. So TriCor Reference Lab is a regional reference laboratory in the state of New Mexico. We provide laboratory services from the most esoteric laboratory services to the most routine services. We're found in hospitals and physicians' offices throughout the state of New Mexico, and we cover all of the types of services: microbiology, clinical chemistry, oncology, and many others. A little bit about New Mexico. I usually have a map of the United States just to orient all of us, because we get interesting questions in New Mexico. But just so you know, this is in the United States. Don't laugh, it's true. But I trust all the laboratorians know this. So our laboratory, we're the sole source laboratory provider for over 9 hospitals in the greater Albuquerque area. This includes an academic medical center with a cancer center, children's hospital, trauma center, all of that. We've got a heart hospital, we've got community hospitals. These are the places where we are the sole source laboratory provider. And then throughout the state of New Mexico, in rural New Mexico, there's 5 hospitals for which we are the sole source laboratory provider. Then throughout the rest of the state, we provide reference laboratory services, which range from 1 or 2 samples a month to a few hundred, up to a couple thousand samples per month from each of the specific sites. So we provide a lot of services throughout the state of New Mexico. And just for reference, New Mexico is the 5th largest state in the union. However, there's only 2 million people in the state. So there's like more than 2 million people in a couple square miles in California, right? So we face huge issues around specimen transport, availability, and then just around acquisitions. New Mexico is also a fairly underserved, resource-limited state. So we just don't have a lot of money for a lot of our laboratory operations, and we have to figure out how to optimize and make the best decisions we can. So when we look at laboratory acquisitions, we have to look at what's getting funded in this current laboratory landscape. And there's 2 models that you can look at. There's one that's a cost-driven model where you really look at return on investment as the primary driver. This will include things like your cost per test and CPT codes and reimbursements, and looking at just the prognostic accuracy, right? Just looking at, what's the sensitivity, specificity of the test. And that model was doable in the past few years. But now, as the drivers for quality are really having a bigger impact and are now impacting hospitals from a financial standpoint, we need to start looking at quality-driven approaches when we're looking at new acquisitions. So looking at the impacts of the new CMS measures, the diagnostic accuracy, patient safety, and harm reduction are all really important impacts that we didn't really have to look at before. All you had to say was, this was my ROI, I think I can make some money, here's the CPT code, we'll get reimbursed, and so give me my instrument. Now there's many more factors that we need to start to include in our ROI studies, but the question really is, how do you look at quality? How do you quantitate these qualitative metrics? Everybody knows how to do an ROI, and that's easy, and I can give somebody a number. How do I put a number on these qualitative metrics? So I didn't go to school for an MBA. I didn't really want to care about most of these financial terms. I did that on purpose because I didn't want to do it, and I was hoping somebody else would just do it for me. But I had to start to learn some of these terms, right? So I have the term and the technical definition, and you don't have to take offense to it, but it's my own personal definition for dummies. You can use it however you need to use it. Maybe you're okay with the technical term. I know I need the definition for dummies when it comes to this, right? So return on investment, the percentage gain or loss on an investment. So ROI, what's the rate of return? Think of investment. You put money in, how fast am I going to make that money back? Okay, that one is fairly easy. We've all done that. The payback period, how long until I get my money back? This is similar to the return on investment, but it's the amount of time until the initial investment is recovered. The net present value, I think, is really fascinating. So it's an estimate of the profitability of a project, but it also looks at what's the present value of this money if I were to just invest it in the bank. If I were to take this money and put it in my savings account, what is the value of this money over 3 years? And what's my savings account interest rate? Will I be able to make the money back better if I just put it in my savings account versus investing it in your project? All right? And then the internal rate of return is used to compare two different projects. Which of these two projects will be the one that makes more money? All right? These are terms I didn't want to have to learn, but it's been helpful for me to at least be around people that know what they mean. So the ROI, we have the financial people, they understand that, they do that, but now we need to really start to look at our clinical justification, right? So looking at our clinical justification terms and definition, there's a wonderful acronym that I think we can really start to use when we're looking at some of these justifications, which is STEEP, looking at safety, timeliness, effectiveness, equity, efficiency, and patient-centeredness. As we're looking at our new acquisitions, if we can start to take these factors into account, we can find better ways to justify the instrumentation that we're looking for. So there's some of these measures that are quantitative, even within some of these quality metrics. So looking at items like turnaround time, length of stay, defined daily dose, the incremental cost ratios, all of these are ones that you can measure, you can put a number on, and you can include that in your ROI. But some of the qualitative metrics, even within these quality factors, how do you really quantify harm reduction? How do you quantify safety? It feels like it's going to be a more safe test. We're not doing as many manual processes. It's more automated. Individuals aren't handling open tubes anymore. A system is opening tubes for them. You know that it adds value, but how do you quantitate the value that it's adding to your system? Provider satisfaction. How do you quantitate that this physician group needed this result, and so it's going to make them, they'll be able to do their job better. How do you quantitate that? And then patient satisfaction is something that hospitals are really, really strongly driven by based on the CMS criteria. So now how can we in the laboratory start to impact the patient satisfaction metrics that hospitals are under and hospitals are facing? So when we're looking at aligning cost and quality, we have this instrument, like Omai said, your new instrument A, and you think it's the best thing. We know we need it. How do we convince administration to purchase it? What we use at Tricor is the business value model. It's a model that takes into account the return on investment and all of the financial metrics that the financial groups are used to using, but then it also incorporates the purpose alignment model, which is basically a strategic justification for why you want to utilize the instrument or purchase, make a new acquisition, and then they'll bring in some other considerations as well. So we balance both the financial as well as the strategic alignment. So how do we do that? We have a basic ROI, and everybody has their own way that their institution accounts for ROI. But I do want to point out some things that we often forget about when we're making our ROI strategies. Okay, so this is just an example where you have your manual, old way of doing things, however you were going to do that. We'll always include that. Then we have our proposed new way that we're going to do things. Always make sure that you include your installation cost in the project around that if you have to do any construction or anything to help to prepare your space. But when you look at the savings that your facility is going to begin to see, always take into account some of the savings items that we tend to forget about. One thing, if you're going from manual to automated processes, don't forget about the cost of repetitive motion injury at your institution, and not only the cost of the injury, but the cost of the premiums that your hospital or your institution is facing. We were seeing increased repetitive motion injuries that actually translated to an increase in the insurance premium for insuring our organization. And if we could bring down the number of repetitive motion injuries, we could actually impact not just the cost of the injury, but the ongoing cost of the premium that the facility was having to incur. So there's lots of opportunities for finding savings as you're making your ROI, because we need to drop some things down to the bottom line. So we understand, economic justification, but what I want to spend the rest of the time really focusing on is how do you align the qualitative and the strategic markers, right? How do you put a number on the thing that you can't put a number on? It would be easy when the ROI lines up and everything is nice. The question is, what do you do when you can't quite put a number on improved satisfaction? You can't quite put a number on harm reduction, or you can't put a number on safety. How do you really quantitate that in a way that you can present it to your committees? So what we use is the purpose alignment model. It's a 2x2 table. Every laboratorian can understand a 2x2 table, right? So we have the market differentiating, increasing market differentiating factors that's plotted against the mission critical nature of a test. All right, so if we start in the upper right-hand corner, a system that's differentiating, it's better than, it's a win-win, we're gonna gain market share, we're gonna get that $1 million increase, we know that it's a win-win, everybody should do it. It's gonna be highly market differentiating, highly mission critical. As we go to the left, there may be items that are not quite market differentiating, but they're mission critical. So that's parity. It's something that's, it's as good as, it's the cost of doing business. It doesn't matter. Maybe that's the refrigerator and that's the centrifuge. That's just, we have to get this done because it's required. It's not market differentiating. Nobody's going to come, to our hospital because we have a new centrifuge, but we need to have it so we can process our samples. Things that are not mission critical, but are highly market differentiating, these are things that are unique, they're interesting, they're curious. Sometimes us lab directors have these fascinating ideas that we come up with in the middle of the night, and we think it's the most interesting thing in the world. And 4 people are going to order it every 12 years, right? Not necessarily the thing that we want to do. It might be market differentiating. It's not mission critical. That's where you look to partner. That's where you look to find a send-out facility to work with you on doing that testing until it becomes more needed in your area. And then things that are neither market differentiating nor mission critical. That's trivial. Just don't do it. If you find yourself in that box, run and don't look back. So how do we apply that to our qualitative metrics? I have listed here a couple of the qualitative metrics that we look at. So we look at quality, improved in-product quality, reduced errors, safety, procedure enhancements, the ability to kind of do things better, having an audit trail, right? When you go to an automated process, you have an audit trail, and now you can actually track and make sure that every sample is connected to every result. Having some improvement in turnaround time, flexibility. Have you ever had a challenge justifying a new instrument, and you say, well, I know we're only talking about one test on this new instrument, but it can actually do 12 others down the road. How do you justify that these 12 others are now going to make sense, so we should purchase the instrument for this? You can actually use these qualitative metrics to say this flexibility is actually a differentiator for us. And then you can add in other quality metrics, other metrics in the system. And so then what we do is we score each one of those metrics using that purpose alignment model. Things that are differentials, differentiators, parity, partner, or trivial. It really forces us in the laboratory to think about each thing, and does it make sense? Is it really needed? And then what we do is we plot this, and this is from a published study in Lab Autopedia. We plot this across the economic justification index, the strategic justification index, and it helps us to see within the lab, have we been able to create a compelling argument for the purchase of this new instrument? It's the first thing that we really do to start to look at, did we make a right decision? And if not, we need to go back and reevaluate. Do we need to identify new partners? Do we need to identify new stakeholders? Do we need to reassess our mission aligned with the strategy of the organization? And so here's just kind of a couple of examples of where things may land. All right, so if you have something that has high strategic alignment, high financial alignment, go for it. That's the thing that has a 1-year payback period and it's aligned completely in the strategy, you absolutely do it. If you have something that's high strategic alignment, but it has moderate or low financial alignment, you can get your quality department or other institutional departments to support you. If you're doing a GI test, you pull in your GI providers and say, we need you to provide a letter of support. We need you to help us to get aligned with this. If you have something that's low strategic alignment and high financial alignment, you need to really think about, do you really need to do this? Just because it's cheap to do, it may not align with the strategy of your organization. So maybe you don't really need to do it. Maybe you need to look for something else. Just because things are cheap doesn't necessarily mean that they're appropriate. And if you have something that has both low strategic and economic justification, again, run, don't do it, don't look back, try again. So just plotting a couple of our experiences with the business value model so you can see where some things landed. For us, one of our differentiating items that was high market differentiating, highly mission critical, but super expensive, and we had to go through a lot of justification to get it, was microbiology automation. We had to go through a lot of work, a lot of justification over time to get it, but we were able to achieve this acquisition because we aligned it to the purpose and the mission of the organization. It was market differentiating, and it was mission critical. Something that we actually identified for us in our institution as parity but moving towards differentiating was doing some increasing automation in our molecular infectious disease section. We knew that we needed to do the more automation in this area for workflow. However, it didn't really change the result. The result wasn't necessarily better. We just had so much volume that we needed to get workflow. We knew that the clinicians weren't really going to care if our workflow improved, and if we had less overtime, nobody outside of the laboratory cares for that. So that was just the cost of doing business. We had to do it because it was the cost of doing business, and it was what we needed to do to survive. So it was mission critical that we got it done, but it was not necessarily market differentiating. It didn't change anything for us. In an area that we had decided that we needed to partner, our courier system was something our laboratory gets all of our samples via courier 24/7. We had our own cars. We had a whole fleet of vehicles. We had a whole automated car system and GPS trackers. And it just became something, we're laboratorians in our organization. We don't manage car fleets. And as that became larger and larger, we realized we needed to partner. It was market differentiating because we had to get the samples to our organization, but it was not our core value. It wasn't what we did well. So we found somebody who did it well, and we partnered with them so that we could get it done. And then things for us, I can't see it. What does it say? Somebody? Oh, okay, so forensic toxicology, for us in our laboratory, once again, we're a clinical lab. We didn't have the space, we didn't have the change control and the tracking control and the handoff control. Like, it was something that we just could not continue to manage within our laboratory for drug screen testing, for pre-employment screening and all of that. Within our facility, the management of that just did not any longer align with the mission and the vision of where we were going. And so we decided this isn't something that we're going to do, somebody else is going to have to do it for us. So these are just a few of the examples of how our items have landed on the business value model and how it's really benefited our laboratory and given us a very clear and strategic insight to when and why we make the decisions we make from a financial standpoint. So just a couple of take-home messages. There's really no single way to do a financial justification. Every facility has their own drivers. Every facility has their own committees and requirements. So it's really not about how you do it. You just have to figure out what works for your organization. And the dollars, though, only make up a portion of the story. It's really important to figure out how to incorporate qualitative metrics into your ROI so that you can do a better job of getting the instrument that you're interested in. You need to learn the mission and the vision of your organization and put your strategy in their words. I love to feed back my CEO's mission with my instrument in place of some of his words. So when he says the goal of our organization is to improve the health of New Mexicans with the right test at the right time, giving the right result, I love to say the goal of this purchase and acquisition is to improve the health of New Mexico by giving them a blah, blah, blah result by a blah, blah, blah instrument, which is going to be at the right time because of this, this, and this. I'm going to feed him back his words, and it makes it really hard for him to disagree with what he's already said. Okay? And then you have to get a seat at the table when if you can, get a seat at the table when they are defining the mission and the strategy so you have a head start and we can start to make sure that laboratory isn't excluded from laboratory decision making. The final thing is getting finance and clinical teams on the same team early to work on a common goal. I always like to put it this way. When I'm looking at a major acquisition that's going to cost, and I know it's going to be a difficult strategic alignment, it might be a difficult financial alignment, and I know it's going to cost a lot of money, I need to get my financial team and my administrative team on my team and not on the opposition. So I look at it as playing doubles tennis rather than singles. When you play singles, it's the lab versus administration, and we're batting the problem back and forth. When you play doubles, you're both on the same side of the net, and the problem and the instrument is on the other side, and you work together to accomplish your final solution. So really, you have to get your administrative team and the laboratory team on the same page to make the final decision. And you really, when you work as a team and not as adversaries, you can really achieve the best outcomes. So thank you.
Okay, so I just wanted to thank Dr. Colbert for her excellent presentation, and we're going to begin the Q&A. So if you have a question, I guess there's going to be a microphone that goes around. I'm not sure who's going to be controlling that. Okay, wonderful. There's a microphone, and yeah, we'd be happy to take any questions that you have.
Thank you. I think an important I sit on the opposite side of you. I make those decisions for my organization. And one of the important things I think that you didn't emphasize is honesty. You can say in your slide, we know we need it. Now we have to convince administration that we need it. Well, if you think you really need it, you should have no problem convincing administration of of your need for it. But the old philosophy of fool me once, shame on me, fool me twice, I'll never approve one of your requests again. So if you're going to do something like this and you're going to come in front of me, you better not be trying to pull one over on me, because I'll find out. And if you tell me that you're going to save $1 million by having fewer cases of whatever it was that you talked about in there, 2 years down the road, I'm going to call you up and ask you, how much money have I really saved? And if I don't save that, I'm not going to be happy.
I absolutely agree. I absolutely 100% agree. And I think it's a place that we often can get ahead of ourselves in the laboratory. If we build it, they will come. And now we project volume increases because of this new amazing test just to get it approved. And so that's one thing that I'm actually really strict on, is I will only do projected volume or revenue or what savings based on current state. And if we're replacing something, I'll pull other volume in, but I won't make volume increase changes. And then I know if I've made a statement around savings, then I have to have a plan to measure it from the beginning. Because I've actually been called into the boardroom to say, we've given you X amount of dollars for X instrumentation, now prove it. And the great thing is, when I was able to walk in and say, this is what we did, this is how we did it, here's the proof, here's the value, all of the sudden, I mean, I don't know what the opposite of the fool me once, it was kind of like, well, here's more, right? It makes it a lot easier down the road to gain trust. We really have to be honest. I totally agree.
So just one other thing, well, two things. The first is exactly what you said, so I actually commend you for that, and I'm going to tell you about something else as well, but you know that if you've come to me and I've approved something 2 years down the road, I'm going to come to you and I'm going to say, where's my money? I want that money. If you told me you're going to save $1 million, I want to see that you saved $1.5 million. I also want to commend you for what you said, your ability to say to yourself, this is not a business we should be in. And your decision to work, I assume, with an expert in courier is to be applauded, because let's face it, laboratorians really don't know much about courier systems. So I think that was a very good decision, something that I hope your CEO applauds. Thank you.
Yeah. And again, it was honesty, right? We had to be honest and say, yeah, we're not good at this.
So I'm the sales rep of that incredible product that you showed in the first slide. So assuming that's the product, from the time that you said, yeah, I want to use this, to the time it's actually used in your clinic on a regular basis, how long should I expect so that when I tell my boss that, yeah, we have a customer, what is that, when are they actually coming online?
I think that's a great question. I think it's going to depend on your organization. Things may move a little bit faster for you at Tricor than they do at UCLA. We are a slow-moving institution. I'm going to be fast to evaluate, and then the next process can take however long it takes. And so what I ask for manufacturers to be is patient in that process, because we're working as hard as we can to do it right. But honestly, emergency situations come up. And so the new best assay method in the world may be top of my list, and it was until I had to build an isolation lab for Ebola, which was a surprise. So then everything else got pushed down the list. So it's really hard to predict, 3 years, 1 year. I don't. It's hard to give you just an exact number. It kind of depends on what it is.
No. Well, and what's interesting about our organization is that we are a reference laboratory, but we're also a sole source laboratory provider, and have a very interesting relationship with what's termed as our sponsor organizations. So there's hospitals that we have to go to the committee, not just the one VAC or other type of committee. We have to go to multiple committees across multiple hospitals and get approval, and not every hospital is going to be aligned with what they need. Right now I have one hospital that's looking to get revenue. I have another hospital that's looking for cost savings. And so I, though, have a philosophy of standardization in my laboratory because I have one laboratory, not 10. So how do I then say this is the next best assay? It's the one that I believe is scientifically accurate, it's clinically important and clinically relevant, it is the right test for our laboratory and our workflow, and I now have to go present 2 completely different business cases or more to get approvals. And those meetings are not always synchronous. I can't always get on the agenda the next time. So, I mean, it's complicated, it's complex. Be patient. We apologize. On behalf of laboratorians, we're sorry. We know you want your bonus.
We want to do your tests. We do. We want to do
We're
your
sorry.
tests.
So this is Domai. So you said $1 million, right? That's what you have to come up with savings. So you have multiple departments. You have pharmacy, you have your surgeons. They're all saying they're newfangled tests. They're all going to save money. And so let's just pretend that, pharmacy, your surgeons and lab is saying, I'm going to save $1 million in the ICU. How do you distinguish who saved $1 million? And that,
you make a great point.
And I've had the same conversation with length of stay. So I presented my test. I said, I'm going to save you 3 days of length of stay. And I had the board member turn around and say, for the amount of people that have come in and given me this, I'll see 3 days of, save 3 days of length of stay, I'll have nobody in the hospital, right? Because everyone's saving 3 days or 7 days or whatever. So how do you distinguish a lab test from another department?
I mean, I think that's a great question. You can't disconnect the interconnectedness of the hospital system, right? And so even though I saved, let's say, isolation room or whatever, there are a lot of other things that are going into that. So let's say something else that year also was saving isolation room days, right? So the challenge then is that as I look at my data, how do I prove it was my thing and not something else, which makes it unfair a little bit to just say your particular initiative has to save $1 million. It's why we were a little surprised to hear it, right? Because we had already gone through VAC and everything looked good, and then when the budget came out, our project didn't get funded. So we were like, what happened? And then we were alerted to the ROI committee. So then we put the ROI together and move forward. But I agree, it's a complicated question, and we're already having enough trouble as laboratorians communicating honestly with what it is that we have, and now we have to reach out and get into other areas where if pharmacy has their own initiative and we're competing for the same big box of money, well, then maybe pharmacy has some data to suggest we're not going to quite save as much money as we thought we were going to save lab, because their initiative then could go through. So I don't know. It's a great question, and moving forward, it's something that we have to work out.
I don't know if it's fair for them to demand that you show $1 million in excess revenue, and that's going to hold your lab back from making progress. So I don't know. I'd like to understand why they're doing that to you. The other thing I was going to point out is you may have already understood that, but when you talked about tying your hospital up for 5 years on a lease versus a purchase versus something else, I'm sure everybody realizes the vendors are not stupid. Whether they bring that machine into your lab on a lease, capital lease, on an operational lease, purchase up front, cost per reportable, cost per test, when that machine comes into your office, into your lab, you're paying for it. So if that machine is a $200,000 machine, doesn't matter how, you're going to be paying that money. So you can't get around it. When you make the decision to bring it in, you got to pay.
No, I agree. That's why it really doesn't matter to me whether it's capital equipment, lease, or reagent rental. None of that really. That's just the understanding of getting the price, getting it into the door.
In many institutions also, they will not allow you to do a capital lease because a capital lease and capital money is the same budget theoretically. And if you have to do an operational lease or an operational lease, that will create problems because you'll have the lease for 2 years, and then you'll have to buy the equipment back at fair market value. You end up paying way, way more for it than if you would purchase it outright. So our organization tends never to do an operating lease. Cost per reportable result, cost per test is probably, generally speaking, the best way to go if you don't have capital.
And we find in our institution, so I've been there directing labs now for 6 years, it changes every 2 years. So 6 years ago, they wanted everything on the capital equipment list. Then 2 years after that, it was some kind of lease. Now, reagent rental is really, really popular. So, I think it's just up to the institution.
All geared to keeping the number of requests you have at a minimum, including the $1 million ROI.
Yes.
Just a couple of comments, if I may. I think one of the real important values of what you've presented here today is that, and certainly in my experience, I'm in Canada now, I've worked in the US before, but certainly in Canada, what we saw over time is that the lab did a poor job of this. So finance and head office took that away from many labs and laboratorians, and they started doing that. Unfortunately, because they don't understand the clinical side of it, they actually do a bad job too. So I think it is important that the lab understands this. We may not like it, but I think we need to understand it and do a good job of it. The other comment I'd like your opinion on is, I think it's very important to try and make this information objective, to try and assign numbers and grade to some of these things. But what I have seen on the other side is very, very complex 200-page RFP documents where you grade this and grade that, quantify this and quantify that. And honestly, at the end of the day, you could pretty well flip a coin. The error bars around those estimates, it's no longer meaningful. And so I would caution people that they need to be realistic about this, because these are not real measurements. These are guesstimations many times, right? So I wonder if you've come across that or what your thoughts are about that.
Yeah, so actually our ROI document, including the strategic justification, is just about as simple as what I presented today. It's not, a big, complex, multi-page document, and our CFO is actually very he likes strategic alignment, but he's very pragmatic in what he asks for, what he looks for. The numbers are clear, they're straightforward, I can understand them. I don't like to fill them out, but I can understand them. And it's something that I can walk in and I could present it. So I very much agree with the statement that we need to first be honest with the numbers. And again, I don't like to fudge the numbers. I don't like to put them in and then tweak them a little bit to make it look a little bit better, pretend like we're going to get new volume. Because as soon as you get one new client, you'll lose 3 more, right? So I like to stick with where we are, what we know today, from a financial standpoint. And then from a strategic standpoint, one of the things that I didn't mention, it's really important to decide or to think about the strategic alignment from all of the different customer points, right? Think about things from the laboratory standpoint. It's going to make the lab more efficient. We're going to need, 12 fewer people. And the clinicians don't really care because the result's exactly the same, right? Is that really going to make the difference in your hospital? So you have to really think about it from multiple stakeholders' positions and then make a decision, where does this really fall within our institution? So I certainly agree with that statement. We have to be clear, we have to be concise, and we also very much have to be honest about our strategic and our financial justifications.
Some of the accounting rules have been changing in the last couple of years, and there's been speculations that hospitals will shift how they conduct acquiring equipment to more purchases. I was surprised to hear you say, Dr. Gardner, at UCLA, there's actually a move towards reagent rentals, because we thought the shift was the other direction. Do you see that at TriCore, Dr. Culbreth?
No, we tend to stick towards either capital purchase or something around cost per test, cost per reportable. I think, again, we're fortunate to have really strong administrative staff that look very closely at our contracts, even within reagent rental agreements. If that's the direction that we do choose to go, they have some fancy contracting language around capping the reagent rental costs, so we don't end up paying 5 times more. So they look at projected volumes, look at the cost of the instrument. But right now we are leaning more towards either capital purchases or a cost per reportable, which is actually really important to take out some of the QC and the proficiency testing and training and all of that, and really focusing on the reportables. So that's the direction we're going in our organization.
I think for us at UCLA, at least within the last 2 or 3 years, we just got bumped off the capital equipment list a lot. And I think within that, now maybe we're just on an unlucky streak, right? Maybe there's just been a bunch of freezers to go down, and that is what it is. But I think that also with a lot of this instrumentation, it's very high capital cost instrumentation. You know, like, wonderful, the machine costs $500,000. We're probably not going to be able to strong arm our hospital for that no matter what it is that we do. And so, you know, we have to look at these other ways, because as much as we love the newer and new platforms, being a relatively high volume, nothing like my colleague here, but being a relatively high volume for an academic medical center, we're looking at big instrumentation, and I think that's part of the issue too.
I'm at the South Carolina Department of Public Health Laboratory. I presented a business case to our Public Health Administration to show a savings of roughly $200K a year. They quickly came back to say, okay, well, what's that going to cost to the other parts of the agency? So I think the hospital perspective is really important, because, and it's just really interesting to see that whole spectrum analysis does matter. So I'm just saying that to other people in the labs here.
Okay, so it looks like that's all the questions. So this concludes the educational portion of the event. We want to thank everybody for attending. Hope you'll stay a while, enjoy the refreshments, network with your colleagues, and Chris and I will be here if you guys have any further questions. So thank you so much.